I'd like to first thank the organizing committee and Dr. Pate and also Dr. Blair and the rest of the committee for inviting me to describe some of the studies that I'm working on at the University of South Carolina School of Medicine, also in association with the Doran VA in Columbia. So this is actually a cartoon that I'm surprised hasn't shown up yet. This is the obesity trends among US adults beginning in 1990. And you can see from the most recent map in 2013 that there's a clustering of these highest obesity rates in the states. And in fact, if you look at this, it's very appropriate that this clustering takes place here in the Southeast. And this is the reason why we're here this week, to talk about these issues, and in particular, to talk about these issues in terms of complications associated with obesity. Now, we are very familiar with the peripheral complications of obesity. Again, I think this is something we'll discuss a lot this week. But one of the areas that I think is somewhat perhaps undervalued or underappreciated are the neurological complications of obesity, and in particular, how obesity can impact the central nervous system. And as was just mentioned, one of the things that my laboratory is interested in is this concern of neuroplasticity. And I define neuroplasticity as the ability of the central nervous system to respond to changes in the external or the internal milieu. One of the more neuroplastic brain regions that we know of is this region called the hippocampus. This is a cartoon of the rat brain, and shown in blue there is the rat hippocampus, and in particular several regions which we'll talk about, including the CA1 region, the CA3 region, and the dentate gyrus. One of the reasons my laboratory is particularly interested in the hippocampus is because this is a well-known integration center for learning and memory in the mammalian brain. And in terms of obesity, we know that one of the hallmark features associated, or one of the hallmark neuroplasticity deficits associated with obesity is impairments in cognitive function. And we know this is true in animals, and we also know this is true in humans as well. So one of the questions we're thinking about is, was mentioned earlier, what are sort of the molecular cellular underpinnings responsible for deficits in obesity, is what are the molecular cellular underpinnings which contribute to these neuroplasticity deficits, to these cognitive impairments in obese animals. So this cartoon here summarizes a lot of work from a lot of different laboratories. And one of the hallmark features that is often associated with neuroplasticity and obesity are neuroanatomical changes, whether that be changes in spine density, whether that be changes in neurogenesis in the dentate gyrus, changes in circuitry. So whether that be circuitry into the hippocampus, circuitry out of the hippocampus, or circuitry within the hippocampus. And generally speaking, what we're thinking about is changes in synapse formation, reorganization of synapses. And these types of neuroanatomical deficits probably contribute to electrophysiological deficits, such as in long-term potentiation. Long-term potentiation is this electrophysiological measure, which is believed to be a cellular correlate of learning and memory in the mammalian brain. So these are sort of some of the, I would say, consequences of obesity. And one of the things we're also interested in is identifying the causes of these complications. So for example, signaling, whether that be decreases in insulin receptor signaling or leptin receptor signaling. So I'm sure we'll hear a lot about, and it was just mentioned in the previous session, about leptin resistance. We know that insulin resistance and leptin resistance are key features in peripheral tissues in obese individuals. But we also know that this insulin resistance and leptin resistance also extends to the hippocampus as well. We'll hear later in this session a little bit about mitochondrial dysfunction and increases in oxygen, reactive oxygen species. Again, well described in peripheral tissues, but we know this also occurs in the central nervous system as well. And an emerging important player seems to be neuroinflammation, increases in pro-inflammatory cytokines. So collectively, all of these types of deficits in neuroplasticity, if you begin to group them and correlate them with behavior, you can see that these deficits in plasticity are associated with deficits in performance, okay? So again, excuse me, all of these data are based on animal models, and there are a variety of different animal models which have been used. I'm sure this audience is familiar with the high-fat diet models, as well as OB-OB or DB-DB mice. We've performed studies in collaboration with Carol Greenwood and Gordon Winokur at the University of Toronto to demonstrate that obese sucker rats exhibit deficits in hippocampal-dependent behaviors that are associated with hippocampal insulin resistance. There's also a lot of molecular approaches, some of which we've just heard a few minutes ago, one of which includes decreasing insulin receptors in the brain, or the NERCO mice. One of the problems associated with some of these molecular approaches is that normally they target all receptor populations in the central nervous system. And one of the things that we were particularly interested in looking at was, what if we could discretely target different insulin receptor populations? So one of the ways that we did that was through the development of an insulin receptor antisense construct, which we packaged into a lentivirus. And now we could specifically target different insulin receptor populations to see if there was functional heterogeneity of insulin receptors in the CNS. So in the interest of time, I'm summarizing about 10 years of work into 5 bullet points. So what we're looking at is if we can specifically target insulin receptors in the hypothalamus. And the reason we targeted those receptors is because of their well-known role in body weight, metabolism, and food intake. We can see that these animals exhibit increases in body weight and adiposity, increases in plasma leptin levels, plasma triglyceride levels. These animals exhibit hypothalamic leptin resistance. And in collaboration with Bob Sherwin's lab at Yale, we were able to demonstrate that these animals also exhibit hepatic insulin resistance. Okay? All of these types of changes take place in the absence of changes in hippocampal insulin receptor expression and signaling. I need to emphasize that because of some of the things I'm going to talk about in a few minutes. So, one of the things obviously we were really interested in, as I just mentioned, is if we're able to develop this obesity phenotype, how will this impact synaptic plasticity in the hippocampus? Here's one example of those neuroplasticity deficits that I was referring to. This is a study we performed in collaboration with David Mott at the USC School of Medicine. And what we're looking at is long-term potentiation. So what we're doing is we provide a high-frequency stimulation to Schaefer collaterals, which project onto CA1 pyramidal neurons. And what you see is you see a long-lasting increase in synaptic strength between these CA1 pyramidal neurons in control animals shown in the black triangles. What also should be very obvious here is in our obese animals, where we fail to elicit long-term potentiation in these animals, okay? So this is an example of an electrophysiological plasticity deficit in the hippocampus of an obese animal. We believe that these deficits in electrophysiology are probably related to changes in the phosphorylation state of glutamate receptors in the CNS. We've also demonstrated that there are morphological changes and changes in synapse formation in these animals. And most importantly, once again, we can show that these animals have deficits in hippocampal-dependent behaviors, okay? So obese phenotype produces neuroplasticity deficits. One of the other areas that my laboratory is really interested in investigating is this idea about comorbid depressive illness and obesity. So the clinical data clearly indicate that people who are obese have a 2 to 3-fold increased risk of developing depressive illness. And those obese individuals that do develop depressive illness are somewhat refractory to normal pharmacological approaches in the treatment of depression. So one of the things that we wanted to demonstrate in these animals is that they also will develop a depressive-like phenotype. I always have to do that in air quotes, okay? Because as we know, rats really don't develop depression. But one of the ways that we can measure that is through a sucrose intake test. And as you're probably aware, if you provide rodents with a choice between water and a 1% sucrose solution, under normal circumstances, they will prefer to drink sucrose. And that's shown here in this blue bar. But what you can see in our obese animals, these animals exhibit a significant decrease in sucrose preference, okay? More simply, these animals are exhibiting anhedonia, all right? So beyond this particular behavioral measure, we've also seen in the 4 swim tests that animals exhibit behavioral despair, and in the elevated plus maze that animals exhibit anxiety-like behaviors, okay? So I think one of the things that we're starting to identify is that there are particular links in the chain between obesity and neuroplasticity. We've identified those links, and now one of the things that we're obviously interested in, beyond identifying these links between obesity and neuroplasticity deficits, cognitive deficits, and this increased risk for depressive illness, what are the ways that we could potentially break the links in this chain, okay? One of the ways that we've begun to do that is by looking at a variety of different food restriction studies, okay? So let me take you through for a minute how we perform these food restriction studies. So we had 3 different cohorts of rats. We obviously had a control group of animals. We had 1 cohort of animals that received the insulin receptor antisense construct in the hypothalamus and was provided ad lib access to food. We had a 2nd cohort of animals in which we began a mild food restriction paradigm immediately after the administration of the lentivirus. This is a group we're referring to as a prevention group. And we had a third group of animals in which we initiated a food restriction paradigm after the development of the obesity phenotype. And we're referring to this as a reversal group. And here are some of the data. So you can see shown in red are the ad lib insulin receptor antisense treated animals. You can see these animals exhibit significant increases in body weight. Shown in blue, which are essentially superimposed with the control animals, are the animals in which we began the food restriction paradigm almost immediately after administration of the lentivirus. So you can see that we're able to prevent that increase in body weight, which was induced by decreasing hypothalamic insulin receptors. And shown in green is our third group, in which we allow the obesity phenotype to develop, and then we initiate a mild food restriction paradigm. And here you can see in these animals, we're now able to return body weight levels back down to what you would see in control animals. And what I mean by, what I mean by a mild food restriction paradigm, we're talking about a situation in which animals are deprived about 20% of their normal food intake. Okay, this does not constitute a stressor for these animals. Okay, so beyond these changes in body weight, we're also able to demonstrate that these food restriction paradigms can prevent and reverse the increases in body fat, as well as plasma leptin levels, as well as plasma triglyceride levels. So these food restriction paradigms are able to prevent or reverse the metabolic and endocrine phenotype associated with obesity. So now the big question is, if we're able to reverse the endocrine and metabolic phenotype, can we restore neuroplasticity in the hippocampus of these animals? So once again, this is the example of long-term potentiation, that electrophysiological correlate in the central nervous system. So what we're looking at in the upside-down filled triangles, these once again are the insulin receptor antisense treated animals that are provided ad lib access to food. These animals have developed the obesity phenotype, and we can see that these animals once again fail to develop long-term potentiation. But you can see the other three groups are all superimposed on one another. Essentially what we've done is by restoring the appropriate endocrine and metabolic milieu, we can restore long-term potentiation in the hippocampus of these animals. We believe that at least in part, the restoration of this long-term potentiation is due to the restoration of the appropriate phosphorylation state of glutamate receptors in the hippocampus. And once again, as I mentioned, there's an important and emerging role for neuroinflammation as it relates to neuroplasticity deficits in obesity. And so one of the things that we've also looked at is pro-inflammatory cytokines. And as shown in this slide, we're using IL-6 as an example, that obesity will lead to an increase in IL-6 levels, and that this obesity-induced increase in IL-6 levels can be completely reversed in both the prevention and the reversal paradigms. And again, I'm using IL-6, but we've seen similar changes in TNF-alpha as well as IL-1. In terms of neurotrophic factors in the central nervous system, we've seen that there are significant decreases in BDNF expression in these animals, and again, which can be prevented and reversed by these food restriction paradigms. But last but not least, we can see that the obesity-induced anhedonia can be completely prevented and reversed through these food restriction paradigms. So I think sort of the take-home message from our studies up to this point is, the good news is, as was emphasized in the beginning of our sessions today, is you can prevent these neuroplasticity deficits seen in obese patients, okay? You can also, which we'll focus on in the upcoming slide, is you can also reverse these deficits as well. And so what I've been focusing on today is on diet, but we also know that there are a variety of other different mechanisms which also can prevent and reverse these neuroplasticity deficits in obesity. So for example, an enriched environment. You can provide animals with an enriched environment basically by giving them a running wheel, which goes further to what we're thinking about in terms of the role of exercise, which we'll hear a little bit more about in our session today. Another component of an enriched environment may be sleep, which is another component that we'll hear about in one of the other speakers in this session. Obviously, in terms of pharmacology, this may also play a very important role as well. So in fact, we're engaged in a study right now with Carol Greenwood's lab at the University of Toronto, looking at the ability of rosiglitazone to prevent high-fat diet-induced neuroplasticity deficits. And I think a really interesting and emerging topic is this idea of bariatric surgery. Obviously, this is not a prevention model, okay? But in terms of a reversal, we know that bariatric surgery can oftentimes very rapidly produce appropriate changes in metabolic and endocrine milieu. So the question is, under those circumstances, What about the cognitive deficits that you see in obese patients? What about the increased risk for neuropsychiatric disorders? Can those neuroplasticity deficits also be effectively managed by bariatric surgery? Because the ultimate goal here is to move those red circles higher up on the plasticity scale as a way to increase cognitive or behavioral performance. We want to move from our red circles closer to our blue squares. So that's a very brief overview of the studies I've been working on in my laboratory at the USC School of Medicine. These are some of my intramural colleagues who've been engaged in these studies. It includes, most importantly, Claudia Grillo, who has sort of been the driving force behind these studies. Ongoing collaborations with my colleagues at the University of Toronto, Carol Greenwood and Gordon Winokur, in Cincinnati, Randall Sakai and Steve Woods, at Hopkins, Kelly Tomashiro, and Bob Sherwin at Yale. And of course, last but not least, our funding sources, which include the USC School of Medicine, our USC Research Foundation, as well as the USC School of Medicine, NIH, as well as the Veterans Administration. Once again, I'd like to thank the organizing committee for inviting me to participate, and I'd be happy to answer any questions you might have at this time.
Yes, what about meditation and mindfulness and how that affects neuroplasticity, and how would that affect this situation?
I'm sorry, I'm having the same sort of feedback problem. Could you repeat your question?
Meditation, mindfulness, things like that that have effects on neuroplasticity?
That's a great question. So I think that it might be somewhat difficult to apply that to my particular studies in a rodent, but I know for some colleagues of mine, that is an area that they're investigating, and it very well could be a very interesting approach. One of the things that people are looking at, I often talk about obesity as a metabolic stressor because overall my research portfolio looks at stress in the hippocampus. And so if there are ways that you can reduce stress, whether that be psychosocial stress or metabolic stress, perhaps through those sorts of techniques, that might be an interesting add-on strategy. I don't know if that really answered your question, but...
I know with stress rodents and stress people, that when you've practiced mindfulness, we've changed the humans back. With the rodents, obviously you can't change. We can't teach them to meditate. Mm-hmm. But hopefully maybe you can do that. But, mm-hmm. But anyway, so it seems like that would have a similar effect with obesity with human patients.
I think that that would be a really interesting area. I'm hoping maybe one of my colleagues will mention that.
Thank you. I was wondering more about the pharmacological therapies that you were developing, like the glitazones. Sorry, I'm not sure if I wrote that down right, but like, what have you discovered with that particular area of therapy? So
one of the areas that we're working on now is, again, with rosiglitazone, which is a drug, sort of a first-line drug in the treatment of type 2 diabetes. And one of the things that we're trying to demonstrate is that the treatment with rosiglitazone can either prevent or reverse the neuroplasticity deficit you would see in an animal fed a high-fat diet. But I think one of the key questions we're also interested in is what's the mechanistic basis for that? Because I think that there's some questions about how actually that might be taking place in the central nervous system.
Well, this is the SEC, and so I would like to use a football analogy. And last night, USC came, the University of South Carolina came back from a neurocognitive deficit and ended up spanking Vanderbilt. Your rats, this is interesting because this is very different from some aspects of bariatric surgery. When you restricted the obese rats, after only being exposed to obesity for a certain amount of time, they were able to regain apparently full neurocognitive plasticity. Have you done experiments to ask, how long the animals, or what period of time they need to be exposed and are still able to retain the ability to recover from that, or if after a certain period of time they can no longer recover from obesity and regain the plasticity?
That's a great question. The aim of the original studies And I should highlight that the original studies were forced upon us by Steve Woods. He said, you must do those studies. So those of you who know Steve Woods, you can't say no, you have to do that study. So the goal of those original studies was to see, okay, once the animals develop the phenotype, can we reverse it? We never took it out farther than 21 days, so to speak. But that is a really good question. I think the other thing I should point out is that these animals are on normal chow. They do exhibit increases in food intake, which we think at least is partially responsible for their metabolic changes, but they're not on a high-fat diet. So I think this goes back to another question we've started to scratch the surface at this meeting in terms of diet, not just in terms of increased food intake, but what's in the diet. Okay, thank you.